Hello, this is Dr. Nilsson. Today we're going to be talking about protist evolution and diversity. Here are the learning objectives for today's lecture. Be sure you read through these and you know all of this information. We will also be working on these learning objectives in lab on Tuesday, where you'll actually be looking at many of these types of protists that we talk about today under the microscope. So what are protists? Protists are the smallest of the eukaryotic organisms. They are in the domain Eukarya because they are eukaryotic organisms, but they do not form a unique kingdom like plants, animals, and fungi do. And while we used to put them in this kingdom Protista in the past, we don't do that anymore because we have new information, and that information is DNA sequence information. So now we know that they don't form a unique kingdom, but they are eukaryotes, and they originated with all other eukaryotes through endosymbiosis. If you're not familiar with endosymbiosis, this is the theory or hypothesis that eukaryotic cells arose because one cell was living in symbiosis with another cell, and eventually it enslaved that other cell and made it work for it. And this is the endosymbiotic hypothesis, and this is how we think mitochondria came to being. And this endosymbiotic event, at least the first one, probably occurred about 1.5 to 2 billion years ago. And this gave rise to mitochondria and other types of organelles that we see in eukaryotic cells. There was a second endosymbiotic event that occurred afterwards, and this is when one of these eukaryotic cells that contained mitochondria engulfed a cyanobacteria. And that cyanobacteria was capable of photosynthesis, and it was the origins of the chloroplast that we see in photosynthetic cells today. So that's briefly how protists came to be, but I want to talk a little bit about some general characteristics of protists. They can really vary in size. We have tiny, tiny algae that are single cells. And then we also can have very large seaweeds and kelps that can be over 200 meters in length. So they can be unicellular, but they can also form colonies or filaments. And even though they're unicellular, they still have complexity. So they have unique organelles, they have things like contractile vacuoles, and they have structures like cilia and flagella that help them move and eat. And some also have cell walls, similarly to plant cell walls. Protists also have diverse feeding habits. Different protists get their nutrition from different sources. Some of them are photosynthetic. This is Euglena. This is a photosynthetic protist. It has chloroplasts, so it can make its own food. But others are heterotrophic, so that means they have to take in food from their environment. And this amoeba here is engulfing a food particle via endocytosis or phagocytosis. Others are parasitic, so they prey on a host. They live within a host and they take nutrition from the host. We're going to be learning about some important, medically important protists to humans later on in today's lecture. Other protists are saprotrophic, and you should know what that means because we talked about this last week when we covered fungi. So saprotrophic organisms are heterotrophic, but they have to digest the food on the outside. So these protists will secrete different types of enzymes which will digest food in their environment, and then they will absorb the simpler molecules to use for food and energy. Other protists are mixotrophic. These are really, really cool organisms. So they're capable of photosynthesis, so they have chloroplasts and other light harvesting organelles. And but when maybe there's not adequate light, or if there's food available in their environment, they're also capable of heterotrophy. So they're able to engulf food particles or take in food from other means. So they're really unique in the sense that they have these diverse ways that they can get their food. Life cycles are also diverse in protists, so they often reproduce asexually, so they clonal reproduction, binary fission, but they are capable of sexual reproduction when conditions are unfavorable. And if you think about this, this is like this is an adaptation to life in changing conditions. So if everything's going great in the environment, the protist will reproduce asexually and all the offspring will have the same genes because that works for that particular set of environmental circumstances. But if something happens in the environment, if it changes, if it gets too hot, if it gets too cold, if other organisms move in, and if it's kind of under stress or in some capacity, it'll switch and it'll reproduce sexually. And what this is going to do is this is going to create variation. So some of those offsprings might be better suited to life in those different conditions. Some protists also can form spores or things called cysts, which it's a stage in the life cycle that allows the protist to go dormant for a while, and then when conditions become more favorable, they're able to continue on the life cycle. And in parasitic protists, a cyst can often be how the parasite is transferred between hosts in this dormant state. Life cycles of protists are very complex. Some of them are outlined in your textbook. I'm going to tell you right now, you are not responsible for knowing the details of those life cycles at all. I do want you to know these general characteristics, though, that I just went over with you. Protists are very, very important to aquatic ecosystems, especially. So the phototrophic protists, so these are the photoautotrophic forms or the photosynthetic protists, they are going to produce oxygen because oxygen is a byproduct of photosynthesis. And they're also going to be the primary producers in aquatic ecosystems. They're going to produce the biomass that all the other animals in the ocean are reliant upon, right? So there is a little bit of plant material in the oceans, but most of the primary production, the initial fixation of carbon, of biomass, is going to occur by these phytoplankton. So what they're these produce and that are suspended in the water, and they are going to be the food source for heterotrophic protists, for fish, which of course will be eaten by sharks and other organisms in the food chain. Whales, though, one of the largest animals in the sea, actually feed directly on plankton, which is one of the smallest. So they're very, very important for aquatic life. I mentioned before that we can't fit protists into a single kingdom. They do not form, just protists themselves, a monophyletic group. They're really hard to classify. They're very different. They don't look the same. You have these big algae. You have these tiny parasitic protozoa. It's really hard to make sense of the diversity of the protists. We're getting more and more information, though, as we get more sequence data on protists, and so the phylogeny, the classifications of protists, is constantly in flux right now. Even between textbooks versions, you'll see changes. The important thing to note is that they do not fit into the plant, animal, or fungi kingdom, and they're not a monophyletic group themselves. So they do not all belong in the same evolutionary lineage. The only way we can make them in the same evolutionary lineage is if we include all eukaryotes in that branch of the evolutionary tree. Okay? So that means the only way we can clump them together in a group in one branch is if we also include plants, fungi, and animals as well. So the way that we talk about protists and the way that we classify them is we use something called supergroups. And what a supergroup is a taxonomic group that's below domain. So these are all going to be within the domain of eukaryotes, but above the level of kingdom. So the plant, fungi, and animal kingdoms are all going to be placed in one of these supergroups along with the different groups of protists. So here are the protist groups here. And so this is actually going to be the entire domain of the eukaryotes, okay? So domain eukaryotes, and then you can see we have 5 supergroups here. The first is Archaeoplastida. This is going to include photosynthetic algae and land plants. We have another group here called SAR. This is an incredibly diverse group that actually kind of has 3 other main groups within it. Excavata, Amoebozoa, and Opisthokonta. Opisthokonta is going to also include the animal and fungi kingdoms. So as you can see, if we just wanted to include the protists, so not the land plants, animals, or fungi, we would not be able to do this by snipping the evolutionary tree in one place. The only way we can include all protists is if we snip here at the common ancestor of all eukaryotes. But this is also going to include the land plants, the animals, and the fungi as well. So all these protists here that are listed are not a monophyletic group, that the protists, as we call them protists, are actually a paraphyletic group. This is a figure that's in your textbook that nicely summarizes the 5 main supergroups of protists. You need to know these supergroups, and you need to know different members of them, some of their features, and their relevance to humans, mainly to human health and to what we use them for, and also the relevance to ecology. You do not need to know the details of the individual life cycles of the different protists. You are going to be getting more experience with these in labs. So this seems daunting right now, but after lab, I think it will be more clear. So what I want to do now is I want to go through and I want to talk about some key features of these different supergroups of protists. The first group I want to talk about is Archaeoplastida. So Archaeoplastida is the group that's going to include the land plants. So these are going to be the plant-like protists, okay? They're going to contain plastids. So you see plastid written there within the name. They're going to have plastids derived from that endosymbiotic event with cyanobacteria. These protists inhabit all sorts of environments, oceans, freshwater. So they're the seaweeds, the green and red algae in the seaweed. They can live in snowbanks, on tree bark, and even on various animals' fur, like this sloth here is covered in green algae. Many of the green algae also form symbiotic relationships with other organisms, including plants, animals, and fungi. And you saw an example of this last week when we did our lichen lab. The morphology can really vary. Some are unicellular, so this really tiny protist here, this is about less than 25 micrometers in diameter, and it's a single cell. It contains a single large chloroplast and 2 flagella. This is called chlamydomonas. And so it's very tiny, and it exists in a solitary form, but these algae can also form colonies like this volvox here. So these circles, these spheres, are colonies of algae made up of thousands of individual cells that resemble chlamydomonas. So they form these hollow fluid-filled spheres. The circles inside them are actually daughter colonies that under certain conditions they'll produce these daughter colonies inside, which will then be released into the environment. We also have larger algae, larger forms of filamentous algae like Chara, which it should look familiar since we studied Chara in our first lab when we talked about the basal plants. So Chara is the closest living relative to the land plants. Red algae is another member of this group. Red algae contain additional pigments that allow the protists to capture additional light in deeper seawater. Red algae is also important because it's the source of useful products like carrageenan, which is used as a food additive. You might see it in your ice cream. And also agarose, which we use in lab all the time to run DNA gels. And if you like sushi, the nori, the seaweed wrapper on your sushi, actually comes from a form of red algae. So that's Archaeoplastida, the plant-like protists. All right, our next group is very, very diverse. This is the SAR group. This is going to consist of the stromatophiles, the alveolates, and the rhizaria. I'm going to talk first about the stromatophiles, which are going to be our brown algae, okay? Also diatoms, golden brown algae, and water mold. So brown algae here, if you've been to the beach, you've probably seen this. There's about 1,800 species of brown algae. Most live in colder ocean waters. They're multicellular. Some form simple filaments. Other forms can get really, really long, like kelp. They contain an accessory pigment that is the source of the brown color. Kelp is incredibly important to marine ecosystems. It provides a source of food and also refuge for sea life, and you'll be learning more about the importance of kelp in lab. If you've been to the Caribbean lately or even down in Florida, you may have seen sargassum all over the beach. This is becoming a problem. This is actually a brown algae that thrives in warmer water, and the rising water temperatures are really creating a mess on the beaches down there because the sargassum, we get large growths of sargassum that wash up on the beaches every day. Diatoms are unicellular stromatophiles, and they're characterized by a very unique cell wall made of silica. They have these really beautifully structured cell walls, and they make up a significant amount of sea plankton, or diatoms. You also may have heard of diatomaceous earth, which is the remnants of diatoms, and it's used actually as a pesticide. You can add it to your soil additive. You can add it, and it'll as grubs and unwanted pests in the soil, they'll like get cut up, and they won't like the diatomaceous earth. It's also used as like in filters and as an abrasive and things like that. I just want to briefly mention golden brown algae is a unicellular form, and they're cool because they're one of those mixotrophic organisms, so they have plastids, they're able to undergo photosynthesis, but they're still able to take in food from their environment as well. And they have this unique flagella. They have two flagella, and one of them has these hairs all over it. So those are one more group of stromatophiles I want to talk about, and those are the water moles. These actually look a lot like fungi, but they are not fungi at all. They are protists, and most of them live in the water, and they form these like furry growths when they parasitize fishes or insects, and they decompose the remains. Most of these are saprotrophic, so like fungi, they will secrete enzymes that will digest whatever it is they want to eat, and then they'll absorb the digested materials back in. And if you look, the filaments of the protists actually resemble fungal hyphae. So for a long time, people thought these were actually fungi. Another one that you might not be aware of is late blight is actually one of these water molds. Late blight is what caused the Irish potato famine, and it's actually a water mold that infects members of the Solanaceae family, including potatoes and tomatoes. Okay, the next group I want to talk about in SAR are the alveolates. Okay, so alveolates make up a fairly large group. One of the largest group of them are they're single-celled, and they're going to include the dinoflagellates, the ciliates, and the sporozoans, or apiocomplexans. So I first want to talk about these dinoflagellates here. There's about 2,000 species of them. They're a single cell, and they usually have this like plate of cellulose over them. And sometimes it has like silica embedded into the plates. They typically have two flagella, so there's one here, and then there's one here in this groove. And one important dinoflagellate is called zooantheliae, and these, you can see here, they're living within the polyps of coral. So zooantheliae live in symbiosis with coral, and the coral provides waste products that the algae then uses as a source of nutrients. And the algae provide the coral also with a source of nutrients. So they live in symbiosis together. You may have heard of coral bleaching that's going on. One of the causes of coral bleaching has to do with these zooanthellae not surviving well as the oceans become warmer and more acidic. Okay, another dinoflagellate of note is the dinoflagellate Alexandrium cantonella, which can cause red tides. So there's a problem in Florida right now with some of the red tides. So this dinoflagellate actually produces a really terrible neurotoxin that can cause fish kills. And then when humans eat the contaminated fish, they too can get really, really sick, and it can cause things like respiratory paralysis. So red tides and coral reefs are both are caused in part by dinoflagellates. The other group, another group in alveolates are the ciliates. So this is one of the most complex of the different types of protists and protozoans. They're single-celled, and they have hundreds of different cilia that beat in a coordinated rhythm that helps them move and also allows them to swallow food whole. And they swallow food by kind of sweeping it with their cilia into this oral groove structure, and then they're able to digest it. Some of these also have what are called trichocysts, which are these areas here along the cell membrane. And what happens is when you touch them or change the pH of the environment, they can discharge them. And there are these barbed threads that come out, and they can help capture prey or help the ciliate defend itself. So this is paramecium. We'll be looking at some of these in lab on Tuesday. Okay, the next group I want to talk about is in the alveolates. Again, this is all within SAR, are the apicomplexans. So these are non-modal protists that form spores. They have very complex life cycles, and they are parasitic. Many of them are parasitic to humans. So I want to just mention three of the ones that are not good for humans. One is Toxoplasma gondii. If you've heard of cat scratch fever, that's caused by this apicomplexans protist here. Plasmodium right here. This is an apicomplexan that causes malaria. So mosquitoes transmit this protist to humans, and that is the source of malaria. Another one is called Cyclospora. This is another apicomplexan. It has been known to contaminate water sources, and it can also get on produce and can cause really bad intestinal illness. These all have very complex life cycles. Sometimes they have multiple hosts. You don't need to know those life cycles. Just know that they're very, very complex, and sometimes they can have different types of hosts. Okay, finally, let's talk about Rhizaria. So Rhizarias are going to include the foraminiferans and also the radiolarians. And these are cool protists. They have these really thin pseudopodia, and then they make a unique shell. It's like a glassy silicon shell that's called a test. And then the pseudopodia will extend out of the holes in the test. If you're familiar with the White Cliffs of Dover, the White Cliffs of Dover is actually made from fossilized rhizarian tests. Okay, the next group I'm going to talk about is Excavata. So Excavata is characterized by having a distinctive oral grooves and flagella. They're unicellular, they're motile, and they have diverse feeding habits. Many are pathogenic and parasitic, but some of them are photosynthetic. So one of those here is called Euglena. So it's photosynthetic, it's a freshwater organism, It has this very long flagella and then also a shorter flagella, so it has two flagella. And it has a few chloroplasts in here that allow it to be photosynthetic. So this is Euglena, one of the photosynthetic ones. These are two examples of pathogenic or parasitic excavata. One of them is Giardia. So this is the Giardia protist that sometimes can contaminate water. This is why you're not supposed to drink water up in the mountains. It's passed through like animal feces, and it can actually attach to the epithelial wall of the intestine and get you really, really sick. Another example of a pathogenic excavata is Trichomonas, right here. So Trichomonas vaginalis, this causes the STD trick. And you can see these two flagella, and it's one of the most common STDs, but it is incredibly curable. All right, let's move on to the amoebozoans. So amoebozoans are characterized by having pseudopods, and they're going to include the amoeboids and the slime molds, okay? So pseudopods, you can see here, so pseudopods are these pseudopodia, plural I guess, pseudopodia, are these cytoplasmic extensions, and the cytoskeletons involved in these, but these cytoplasmic extensions that have cytoplasmic streaming allow these protists to move and also to help engulf their food, which again is called phagocytosis or endocytosis. One amoeba of note is this one called Naegleria fowleri. And these, I don't know if you've heard about these in the news, but they're the brain-eating amoebas that can actually be found in warm freshwater ponds. And these are kind of becoming, it's still really rare, I don't want to scare you too much, but the cases of this brain-eating amoeba are increasing, especially in the southern United States. Usually what happens, it happens often in kids. Kids will swim in a warm pond and they'll actually inhale water. And these amoebas will enter in through their nose and then make their way up into the brain, where they will eat the brain and kill the person. And very few people have survived an infection by this brain-eating amoeba. Another member of the amoebozoa are going to be the slime molds. Scientists thought these were fungi, but they're actually not. They're amoebozoa. They're awfully brightly colored. You'll see orange ones, yellow ones, almost like neon color. And they extend their pseudopodia through moist soil, leaf mulch, and rotting logs. And they will digest and engulf bits of the decaying material. And finally, the last group I want to just briefly mention is Opisthokonta. These are single-celled and multi-celled protists that are the most closely related to animals and fungi. I don't believe we're going to be looking at any of these in lab, but they're very complex. These choanoflagellates are actually closely related to sponges, and they form these rosette-like structures. And then the nuclearids here, close related to fungi, and they are saprotrophic. Okay, so that is a brief overview of the protists. We will be talking more about protists in lab on Tuesday. Okay, bye.